Okay. All right. So Beth, Dr. McDougall, uh, glad to really take this time to share because we've had so many people that although there's a good deck and there's some information out there, a lot of it's really technical and they would really just sort of like to be able to hear sort of a visual presentation and your understanding, your relationship with Immuncor and this technology. So look forward to taking this next little bit of time and sharing.
Yeah.
Okay, so maybe we could just start out and give us a little bit of an understanding of the history of this technology.
Okay. So Immuncor's technology is based on the work of Dr. Anton Mehr. Dr. Anton Mehr developed the latest smallpox vaccine that was given. And in the era when smallpox vaccines were being given, it was noticed by doctors around the world that oftentimes if a patient received the smallpox vaccine, they would get better from other things that ailed them. Like if they had a chronic infection, sometimes it would go away. Sometimes solid tumor cancers would improve, autoimmune diseases would remit. And so he decided to devote the latter part of his career to studying that phenomenon. And what he found was that there are proteins on the outside of all of the pox family viruses that will induce an immune modulating effect in humans and animals. And what immune modulating means is that if there were aspects of the immune system that were underactive, it would bring those up. And if there were aspects of the immune system that were overactive, like in autoimmune diseases or in excessive inflammatory processes, it would tone those down. So what we have today is a product based on his work. So he developed a product that contained two highly attenuated and then inactivated pox family viruses. And attenuated means that he took the virus and grew it in various types of animal tissues over many, many years. He attenuated it over 400 times, which is fascinating because if you get a viral vaccine, usually it's only attenuated about 3 times. So 400 attenuations evolved the virus away from being able to infect humans. But then it was interesting because in the end, he inactivated it and used it as basically an antigen product.
You mean he killed it?
Yeah, he killed it.
Yes.
And that's what we have today is we have a product that has two highly attenuated, inactivated viruses that then are killed, and it's given by injection, and it's an antigen product. And so, yeah, so what we have found in 20.
Well, can I ask you a quick question? Yeah. So maybe can you just share a little bit about, although it's not really, we can't really call it clinical data, but some of the work that was done with humans in Germany and the types of conditions that they saw benefit from.
Absolutely, yes. So although it never underwent formal human clinical trials in Germany, it was used in over 1,500 people, and the results were compiled as a series of anecdotes and actually published. And so it was used for a whole wide variety of infectious diseases primarily. So there was a lot of herpes family viral infections, varicella, herpes 1, herpes 2. It was used for influenza. It was used for a typical cold virus. It was used there were a couple HIV patients in the 1,500. And then there were also people with just solid tumor cancers, like some breast cancer, prostate cancer, and someone with lupus, someone with rheumatoid arthritis. So it was a whole spattering of different types of medical conditions, and they saw improvement in all of these. So it significant improvement. So
which is pretty extraordinary to be able to have one thing that had improvement across all those categories. And just one other thing on that, maybe just mention just real briefly about any side effects and the safety.
Yeah. So no, there were no side effects reported, and it was given a safety rating of 1, which is the same as aspirin.
Okay, awesome. Okay, so now I think you're just going to talk about really what motivated you to sort of get involved with this technology.
Yeah, I'm an internist and I've been practicing for 23 years. I have an integrative practice, and most of the conditions I see are immune dysfunction based. I do a lot of multi-infectious disease work, see people with chronic fatigue syndrome with reactivated viral infections. I see a lot of autoimmune disease and a lot of inflammatory conditions, neuroinflammatory conditions, rheumatological conditions, etc. So there's an immune dysfunction basis in all of these things that I see. And I've always been looking for an immune modulator. And really, there isn't anything that exists in Western medicine or even with nutraceuticals at this point. There are things that are called immune modulators, and they block a particular receptor or knock out a particular cytokine or tumor necrosis factor, something to try to blunt an immune response. But there's nothing available that actually balances the immune system. And to me, the immune system is this exquisitely complex, beautiful system that involves multiple cell lines and thousands of intermediates and hundreds of thousands of genes that are transcribed in real time to produce these intermediates. And that takes an incredible amount of coordination, orchestration, and so what I've been always looking for is something to bring balance to the immune system.
And so I think, to me, from what I've heard from people with my absolutely non-clinical skill set, but just from the potential of a product that could be brought to market. So not only is it immune modulating, but it works with the innate immune system because it's non-antigen specific, which I think you're going to talk in a couple of minutes about the effects and the research that's been going on with COVID and where we're going with COVID. But having something that would be not contingent on a variant, it could have this whole wide spectrum of things, would really be critical. So sort of the holy grail of immunotherapy would be an innate immune inducing agent that also is immune modulating.
That is absolutely right. Absolutely
Yeah.
right. And what was interesting is that in about 2017, we brought the product to University of Nevada, Reno, and started working under Dr. Kenneth Hunter, who is a world-renowned immunologist and molecular biologist. And so we started characterizing the product there in his lab and really looking at what it was doing with the human immune system. And we found that the product would induce immune cells to increase their production of interferons, which is these critical information molecules that govern the innate immune response. And then we found also that although it was upregulating these beneficial aspects of the immune system, it was not appreciably upregulating pro-inflammatory cytokines and things like that, compared to other agents that we were comparing it to. And so we had really established all of that, and then COVID hit. And it was a fascinating moment because we realized that we had this product that upregulated the innate immune system and very much upregulated antiviral immunity, but didn't stimulate pro-inflammatory cytokines. So we realized how our product was uniquely poised to address the key challenges of this emerging COVID environment.
And that might even be just worth even commenting on again, because it's so important of what's happening right now.
Yeah. Yeah. I mean, so what right in the early days of, March of 2020, we were just so perfectly suited, because what we were learning very quickly about what SARS-CoV-2 was doing is that it blocked the evolutionary advantage of this virus, is that it blocks the production of intracellular interferons. So let me just back up for a moment and say, normally when the body encounters a virus, viral invader, and a virus invades a cell, immediately that cell begins transcribing genes that code for interferons. And it bumps the level of interferons up, 10x or more. And interferons are secreted outside of that cell, bind to receptors on neighboring cells, basically sounding the alarm that there's a viral invader, and that puts the neighboring cells on alert. And so then if the neighboring cells become infected with SARS-CoV-2, they immediately can block the replication of the virus. There's a lot of other things that happen as well,
Right.
but nonetheless, SARS-CoV-2 is an RNA virus that codes for a family of proteins that actually block the transcription of interferon genes. And then it was becoming apparent in the literature that that initial blockade of a swift immune response is what led to the cytokine storm. It's almost like you need an initial uptick in inflammation in order to stimulate counterbalancing anti-inflammatory cytokines to kind of calm that down. So we were the very first product considered for study when Iowa State University Veterinary School flushed out 2 animal models for studying COVID with applicability to humans. So there were 2 models that were considered applicable to humans, and there was a Syrian hamster model, and we were the first product chosen for study. So what they did is they gave the hamsters a really huge dose of SARS-CoV-2, and then on day 2 and 4, they treated the hamsters with our product via injection. And then we saw a 90% reduction in the quantitative PCR of SARS-CoV-2 in the lung tissue of the hamsters. And then also, very interestingly, when they looked at the lung tissue with histopathology, they saw much less inflammatory damage.
Yeah. Okay, beautiful. So basically, the technologies had recently had one study at Iowa State University, which is the leading veterinary school, and showed this 90% reduction of the virus. And what's happening now with where it's going and the direction in the killed version of this product.
So we're doing a second study at Iowa State right now with a much larger number of hamsters, and we're looking at cytokine profiles and things like that in them. And actually, we should have data rolling in next week on that. So, okay. So that's very exciting. And then we're also doing a study with a contract lab studying the effects of our product against influenza in mice. So there's a mouse model of influenza. And so we're infecting the mice, and then we're using our product, and we'll also have data rolling in the next couple of weeks there. So if we can show the benefit of our product in 2 different animal species against 2 different viruses, that's very powerful.
So if we were with somebody and we were going up the elevator, 15 flights, and we had 1 minute, and we were to say, okay, this is why we believe that this is by far the leading agent available to deal not only with SARS-CoV-2, but with this whole coronavirus issue. How would you bullet point why we should be really considered as the leading opportunity to help support this challenge?
Okay, well, mainly because our product upregulates the innate immune system and places the body in a state of antiviral immunity. It can be used both prophylactically and therapeutically. It can be used as a standalone and in combination with other things. And most importantly, the innate immune system is what we need to be focusing on, particularly with the rapid antigenic drift with SARS-CoV-2. So that virus mutates so quickly. If you are focused exclusively on vaccinations and producing a specific adaptive immune response, by the time you encounter the virus, your immune response is already outdated. So we're seeing this right now with the variants. Like in Israel, for example, 50% of the people that are getting sick with the Delta variant have been previously vaccinated. And so from the beginning, I was concerned this was going to happen, is that the immune system is going to be primed to produce a specific immune response that is not exactly targeted to the viral invader.
Right.
Whereas if you upregulate the innate immune system and you're upregulating the natural kind of way the body keeps the virus in check until it can mount an adaptive immune response that's targeted and specific, you are always going to be able to have a very targeted response to the virus that comes your way. And it's good not only for SARS-CoV-2 and the variants, but for the common cold and influenza and HPV and reducing herpes outbreaks and whatever. So
Okay, so long elevator ride. Okay, but so summary, prophylactic and as a treatment.
Yes.
Potentially supportive of every potential variant.
Mm-hmm.
Completely safe.
Yep.
And extraordinarily economical.
That's right.
Because it has no, preservatives, no, exogenous, DNA.
Kind of grows itself.
It grows itself.
Yeah.
So basically, one product should cover all current and all future potential issues.
Yeah.
It's prophylactic and as a treatment, economical and safe.
And we should talk about the patent that we were just awarded.
Yes, please.
Very exciting, because we've had a variety of provisional patents that we filed, and we finally got one awarded that covers the entire Pox viridae family, and using that against infections that suppress the innate immune system.
Yeah, which
we are realizing SARS-CoV-2 is not the only virus that does this. There are so many viruses that do this. That's how viruses can get a foothold in the person. So rabies and herpes and a variety of viruses do this. So it's a very broad, encompassing patent. So we're very, very excited about that.
Yeah, so you and the team have really described this as a new paradigm, and I think it's going to take people a little bit, even really skilled, really experienced, doctors and researchers in this space, because even our own team, when we first went to them at UNR and Dr. Ken Hunter, they were so hesitant to believe that one agent could do all this. But this, I think we really have to understand, look, this is here, this is real, it's ready to go. We have a laboratory now that can manufacture the product. So really what we're looking for now is a partner that can help get this out to the world.
Yeah, that's absolutely right. That's
okay.
We have a beautiful phase 1 human trial queued up that we produced with multiple consultants, and it took quite a while. And then we have a phase 1B already designed for studying our product against humans that are infected with SARS-CoV-2.
And we've got a couple countries that are lining up that are potentially willing to give us very, very rapid use on humans because of its safety and its potential efficacy, which would allow us to get to market quickly.
That's right. Okay.
And I think probably maybe just one other little part on this before we move to the other aspect of this is that, yes, right now, COVID, the variants are in the collective consciousness of humanity. But then as we start moving out of this into other realms, this can be a complementary treatment to so many things. It can be a standalone treatment. There's one product can really work with all of these things.
And we've also, I want to say, This is another aspect to the inactivated product, had a breakthrough in our in vitro work where we discovered how to basically encase this in an oral deliverable.
Yeah, that's super exciting because right now it's injectable.
Yeah.
And there now is the potential to give it to an oral deliverable with the delivery mechanism that it is attached to would make it even stronger.
That's right.
Yeah, very, very, very exciting. And we can't talk about it because we're just working on the intellectual property now.
Yeah, cool.
Yeah.
Good.
Okay, really beautiful summary. Thank you. So now, so that is a summary of the Immuncor immunotherapy product that was the core technology that was inactivated, but then it is also killed.
That's right.
Now there's another branch that has been discovered for the use of the Immuncor technology, which is, yes, the core attenuated material, but use in its live form.
That's right.
Okay, want to share about that?
Absolutely. Yeah, I mean, we are standing on the work of, on the shoulders of Anton Mehr and all the work that he did. And we never really understood why he bothered attenuating the virus 400 times in so many different animal species. And we used to joke, like, why would he ever do that? Why would he spend so much time doing that and then kill it in the end? Well, now it's turning out to be the most incredibly advantageous thing for the company, because we've discovered that one of the two viruses, when given live, is a powerful oncolytic virus. And so oncolytic viruses is a whole new area of research in oncology. And basically, viruses have a predilection for infecting cancer cells over healthy cells, because viruses don't carry around their replication machinery with them, and they have to infect a cell and hijack the replication machinery of the cell. And because cancer cells are rapidly dividing, they have a lot more of that machinery available for the viruses. So a virus, because of that and other reasons, will readily infect a cancer cell. So this is an emerging vein of research in oncology. And there's a lot of different oncolytic viruses being studied, but there are many reasons why our virus is going to rise to the top and be the best in class, and actually first in class in terms of oncolytic viruses for cancer. So number one, we in vitro in our lab in Reno have already found that our virus will kill 100% of tumor cells in 6 different tumor cell lines in a very short period of time. But the advantage of the attenuation of our virus is that it will not kill healthy cells of the same line. So we found this out kind of accidentally in the beginning, where we studied the oncolytic virus in a human monocytic leukemia line. And then we found 100% killing within 48 hours. And then we tried treating healthy monocytes and no killing at all, and actually added fresh virus to the petri dish every day for 10 days. Even when the cells were old and devitalized, the virus would not infect the healthy cells. So this is very important, because one of the oncolytic viruses that have been approved in the United States is a herpes family virus for treating melanoma. And 2 people died in the trial of herpes encephalitis. And that's because cancer patients are often immunocompromised. They've had often chemo, sometimes radiation, their immune systems are suppressed, and they couldn't mount an immune response to, and so they ended up with like a fulminant infection in the brain and died. So the fact that our virus will not readily, if at all, and we have not found that our virus will infect healthy human cells at all, the fact that it won't is very advantageous for immunocompromised cancer patients. So that's number one. Number two is that we have found that we have learned how to adapt our virus to treating tumor cell lines that other oncolytic viruses will not kill. And so that this is thanks to Dr. Hunter and some really important work that he's done. And so we are first in class in a couple different human tumor cell lines, and we anticipate that we will be for many, many more. And so every time we adapt the virus to a different tumor cell line, that's a new virus. So it's very valuable for the company.
And there's gonna be a little bit more, but let me just summarize on that real quick.
Mm-hmm.
So basically in vitro, in the lab, 6 different tumor cell lines were exposed to our material,
Mm-hmm.
and it killed 100% of all the cancer cells within 24 to 48 hours and 0 infection of healthy cells.
Of the same tumors, of the same line.
Yes, of the same cell line.
Mm-hmm.
And so that's, and then we've created a patent portfolio that's just starting, whereby we can then, we've created a method of adapting our technology to specific tumor cell lines. So the potential for maybe every tumor cell line out there, every type of cancer cell could be, we could adapt it for that particular type of cancer.
That's what we anticipate.
Yeah. Okay. Yeah. Not trivial.
Not trivial. And
okay,
so
go ahead.
We're at the point right now where we are moving to animal xenograft models. So we're now going to be putting the human tumor cell lines into mice without immune systems, and then treating the mice with the virus. So we've queued up a variety of different studies with an independent laboratory, and we're also going to be studying a really interesting marine cancer, so a mouse cancer that is it is the closest experimental model for triple negative breast cancer in humans. So it's a really interesting study that we've queued up there, and we're studying our product with and without, but with and in combination with checkpoint inhibitors. So this is going to be very interesting. If we can show that we can augment the effectiveness of what's now kind of the leading cancer therapy, checkpoint inhibitors, we're going to really have a powerful package to sit down at the table with some biotech partners.
Yes. Okay. And then, so before we summarize, maybe just one other benefit is that with at least so far, every other oncolytic agent, every other virus, the body can build an immune response to it because of what it does to live viruses. However, ours is extraordinarily unique, and that can't happen.
It's really interesting. This is a bit to explain, but the virus that we're using is a parapox virus, and that's the type of virus that would usually give a pox on a sheep. And if a vet was taking care of a sheep that had a pox, the vet could get a pox on their skin. And over many, many years, it's been tried to develop a vaccine to give to vets so that they wouldn't keep getting reinfected with pox on their skin whenever they would come in contact with a sheep. And they were unable to develop a viral vaccine. And so the live pox virus has all of these different mechanisms, like multiple mechanisms that inhibit the human immune system from being able to develop an adaptive immune response to the virus. And so that's very advantageous in this situation, because that is not the case with the other oncolytic viruses being studied. And so when you treat for cancer with an oncolytic virus, it's not a one-time treatment, it's a multiple treatment. And so for most of the viruses out there, it will work maybe one time, maybe less well the second time. By the third time, the person's going to be able to mount an immune response to inactivate the therapy, and they will not be able to do that with this virus.
Okay, so in summary, as an oncolytic agent, we potentially have the most widely applicable technology for who knows how many tumor cell lines, but we already know at least 6, but probably well over 30. Every one of them is its own patent. It is completely safe. So far, it's not been shown to be able to infect a human cell. And because of its nature and some modifications that have been made to it, the body doesn't mount an immune response to it, so it can keep being used as being beneficial. And it potentially is the most economical treatment out there, because again, it's basically just it's growing specific viral material in a specific condition.
Yeah, and probably will be a standalone therapy, and probably will be able to be used in combination with other oncology agents out there.
Okay. All right. So yeah, it's now we're just basically we're looking for 2 things at Immuncor. One is capital investment partners that will help us help the company finish these last studies. And then the animal toxicology study. Once that's done, we've got one country that's ready to start on potentially 300 to 500 people and fast track it. And then these other studies that we've got, and the potential to begin licensing. Today is, it's June, near the end of June 2021. The potential to begin engaging in licensing before the end of the year exists.
Yeah,
that's how far along we are.
We're that close to having a very good package to sit down with biotech partners.
Yeah. Okay. Super great summary and analysis. Beth, thank you so much. And I think this will be really helpful for some people to get a better understanding of what we're doing.
Yeah, my pleasure.
Okay. Thank you. Talk with you later.
Okay.
Okay. Bye-bye.